So today we're going to have a look at the effect of a gene mutation, what an effect a gene mutation might have on the polypeptide and therefore the protein that a specific gene may code for. To start off with, we just remind ourselves what the link between the genetic code and DNA is between the production of a polypeptide and therefore a protein. So on the far left hand side, I've just got an example. We just need some DNA base sequences to work with, some triplet codes to work with. So on the far left hand side, we've got a sequence of, and I'm just keeping it short, 4 triplet codes. You can use your mRNA codon table just to double check that these are the corresponding mRNA codons that you'd get produced during transcription of that particular gene, and then again using your mRNA codon table, you can see that these are the specific amino acids that are coded for by those particular mRNA codons. We know that finally at translation, what actually happens then is you get a specific sequence of amino acids in the primary structure of that polypeptide and then leading to a protein. We've talked about the fact, and we looked at this in our topic of proteins and protein structure, that it is the sequence of amino acids, that primary structure, which then goes on to determine the secondary structure of the polypeptide chain, and then the tertiary structure as well. And the reason why that is due to all the bonds that are taking place between the atoms within the R groups of these different amino acids. Remember, each R group contains a different variable set of atoms, and and ions for that matter, and it's where those bonds take place that folds the polypeptide chain into this very specific 3D shape. The phrase that we use is the fact that this protein is functional. So I've given an example here of an enzyme. Here is the enzyme, here's its active site. The substrate is complementary to that active site. We've described this protein as being functional. So what we need to look at is what is the effect of a gene mutation ultimately on the protein that is coded for by that gene. So we need to know the definition of a gene, and we've discussed this already. A gene consists of a specific sequence of bases that determines the polypeptide chain that is produced due to the specific sequence of amino acids. Therefore, a gene mutation is where we get a change to the sequence of DNA bases within that gene, and potentially then we are going to see the effect that has on the protein formed. There are 2 different types of gene mutation that we need to look at on our syllabus, and they are referred to as what's called point mutations, because we're just going to look at the effect of changing a single base pair within a DNA base sequence, so just one base pair. The 2 different examples are deletion, and you can probably guess what's going to happen here. So one of these base pairs is going to be deleted, is going to be removed, and then the other type is referred to as substitution, and that's where one of these DNA bases is swapped for another. We need to appreciate that these mutations occur during DNA semi-conservative replication that we've looked at previously, and when we have a look at cell division, we'll appreciate that DNA semi-conservative replication occurs towards the end of interphase in a cell's cycle, when it's then preparing for cell division. Right, so let's have a look at an example of a deletion point mutation, and these are just examples, and we need to be able to interpret any example. Okay, so if we have a look at deletion, then you can see what I've got here. Here's the normal DNA base sequence. You can see each of the DNA triplets. So what happens within a deletion mutation is that one of those bases is going to be lost, is going to be removed. So let's just say, and I've just abbreviated just to fit on the page, that mDNA, this is the mutated DNA. An really important point is that students sometimes think that mutation means that the word mutation means a change to the amino acid sequence. It's a slight pedantic point, but mutation only refers to the change to a DNA-based sequence. That can have a knock-on effect on the amino acid sequence, but mutation is referring to a change in the DNA-based sequence. Just a bit more terminology. When we refer to each of these triplet codes for DNA, we know that we've got 3 bases that sit within them. We refer to each of the positions that a base sits in within a triplet code as a frame. So in this case, adenine is in frame 1, guanine is in frame 2, thymine is frame 3 of the first DNA triplet code. In DNA triplet code number 2, we've got cytosine in frame 1, guanine in frame 2, and cytosine in frame 3, and so on and so forth. So I'm going to say then that initially in DNA triplet code number 1, everything has remained the same. So we have A, G, T. What I'm going to say then is that in DNA triplet code number 2, it's the cytosine in frame number 1. So it's the cytosine in frame number 1 that ends up being removed from that DNA base sequence. So the impact on that is that the subsequent DNA bases are effectively all going to move 1 frame to the left. So what do I mean? So the guanine, which originally, in the original DNA base sequence, was sat in frame number 2 of triplet code number 2, that guanine now moves 1 frame to the left. So now we have guanine. The cytosine, which was sat in frame number 3, now moves 1 frame to the left. We now move into what was the third DNA triplet, where we had thymine in frame number 1. Well, now it moves 1 frame to the left. So now it occupies frame number 3 in DNA triplet code number 2. And this just occurs along the whole length of the DNA base sequence. So a nice little phrase to be able to use is when a deletion mutation takes place, we have a frame shift to the left. What you can do now, you can pause the video in a tick. You can then have a go at using your mRNA codon tables. This would be a typical sort of question that you might get, where you then have to work out what, if any at all, what effect this mutated DNA base sequence would now have on the corresponding mRNA codons that are being formed. So if you pause the video, you can then have a go at doing that. In terms of what you get, so remember the first DNA triplet code is unchanged, so we've still got U, C, A, and therefore you'll still get the corresponding amino acid. The issue now is what's happened from the point of the mutation onwards. Because we've had a frame shift to the left, each of these mRNA codons then in this particular example are going to be altered. So we've now got C, G, A. We have got U, U, G, and in this case because I've only got a short DNA base sequence, we pause there, but it still just illustrates the point. So we've now got mRNA codon of CGA and UUG. Again, you can pause the video, use your mRNA codon table, and find out what those corresponding amino acids are. And so they are arginine and leucine. Oops, just need the first three, LEU, sorry. So if we then have a look now, we can draw a comparison between the amino acid sequence within the primary structure. This is when we have our normal DNA base sequence. So we had serine, alanine, lysine, glycine. You can now see that there's been a change to the amino acid sequence within the primary structure. The consequence of that then is that you now are going to potentially get very different sequence of amino acids in the polypeptide chain, and therefore the overall three-dimensional shape of the protein is very different because you're getting bonds forming in different positions. And so in this case, you can see that if I give an example of an enzyme in its active site, the active site is no longer complementary to the substrate. And so the phrase that we need to be able to use is that we now have a non-functional protein. A non-functional protein. The other example of a gene mutation, of a point mutation affecting a single base, is then that of substitution. So substitution. Again, if we start off with the original DNA base sequence, I'm going to say that when the mutated DNA base sequence, the first DNA triplet code is going to remain the same. So we'll have A, G, T. And with the second DNA triplet code, let's say that now we have C, G, and we'll have C, G, and then A. So it's that cytosine which has now been substituted for an adenine. The rest of the DNA triplet codes just remain the same. So we now have T, A, A, and we've got C, C, C. If we then look at the corresponding mRNA, again you can pause the video, you can have a go at doing complementary base pairing and working out what the corresponding mRNA codons would be. So we get UCA. Again, there's no change there at all in terms of the codons formed, and you would get serine as we saw previously. In terms of the mRNA codon, which we now get for DNA triplet code number 2, we now get, get my right colour pen, we now get GCU, and then the other two mRNA codons are the same as previously, so A, UU, and G, G, G. So this then raises a point, and this is a typical question that you might be asked, and it's about the fact that the genetic code is degenerate. So if you remember, what degenerate is referring to is the fact that an amino acid may be coded for by more than one DNA triplet code. And in this instance, again, if you use your mRNA codon table, you will see that alanine is not only coded for by let's get it absolutely right. Alanine is not only coded for by GCG, that's absolutely right, by GCG, as we have in our original, but also it's coded for by GCU. So in this instance, even though there's been a mutation, because the code is degenerate, because an amino acid can be coded for by more than one DNA triplet and therefore mRNA codon, we've got the same amino acid. The other two amino acids are identical. And in this case, in this particular scenario, we've got exactly the same amino acid sequence as we had with no mutation, and therefore we would get, again, a functional protein, no change. That's not to say, though, that a substitution mutation will have no effect at all. It potentially could have an effect. The mutation may cause a change in the in that DNA triplet code, and therefore you may get a change, even though it's just one of the differences between substitution mutation and deletion mutation is that in a substitution mutation it only will affect one amino acid maximum, if at all. In this case, no change. With deletion mutation, because you get a frame shift to the left, it's going to have an effect and probably is going to change all of those different amino acids on from the point of the mutation. So substitution mutation only affects one amino acid maximum, and so it still could, that change could still lead to producing a non-functional protein. But typical questions with substitution are revolving around the fact, just to repeat for the last time, the code is degenerate, and so in this case the same amino acid is coded for. Just to finish off, we have to be aware of the fact that there are certain factors that increase the rate of these mutations. Mutations occur completely randomly all the time when DNA is replicating by semi-conservative replication. However, there are certain factors that can increase the rate at which those mutations occur, and these factors are referred to as, and they are called, mutagens. They have a mutagenic effect. They increase the rate of mutations taking place. Just some different examples that we need to know about are things like ultraviolet rays or X-rays. These are examples of mutagens. There are certain particles that potentially can be mutagenic, so alpha to a lesser extent, and beta particles more so. And then there are a range of different chemicals that can be mutagenic. Things like radon gas, for example, and a whole host of different chemicals.